Yeah, I'm thrilled to be here tonight with you all. I am the Working Lands Research Director, and I'm the Principal Soil Ecologist at Point Blue. For those who don't know Point Blue Conservation Science, we're a nonprofit based just north of San Francisco in Petaluma, and we focus generally on climate smart conservation. And so we've been around for over about 50 years now, and we started as Point Reyes Bird Observatory. So we have a long history of conducting ecological monitoring to inform conservation action, and doing so sort of hand in hand with, I would say, restoration and implementation. So today I'm going to talk about climate smart restoration and how that can help to promote resilient riparian habitats. My outline, so I'm going to talk about three sort of main buckets. So I'm first going to sort of, I'm first going to talk about sort of remind ourselves why riparian restoration is important in the first place, discuss how we can make restored riparian areas resilient to climate change and other stressors, and then think a little bit at the end about how we can harness the power of fungi to promote that resilience. And I have a number of faces up here, and I just wanted to sort of acknowledge up front the work that I will be highlighting tonight is not all mine. It is in some cases very much not mine. It's work that I have the pleasure of witnessing and helping out with, but is led by others. And so it's a collective effort, and I just wanted to recognize the many who have helped to contribute to the climate smart restoration approach that that we have at Point Blue. So why is riparian restoration important? Riparian forests or riparian areas, they're amid these semi-arid and arid landscapes that we have in the Western United States. They provide critical habitat for wildlife and fish. And so they actually can act as like biodiversity hotspots on these landscapes. They provide habitat for endemic species like the California giant salamander, and they act as natural wildlife corridors for mountain lions and coyotes and bobcats that prefer moving along riparian corridors over sort of the surrounding land in the region. They, in addition to sort of providing critical wildlife habitat, they of course support, a healthy riparian habitats support fish like the coho and Chinook salmon and steelhead. And when a riparian area is healthy, it helps to minimize soil loss with, in many cases, what are really erosion-prone landscapes, like we have on the coast. And it helps to maintain soil health for landowners and improve water quality. And more what we're seeing is that, or what we're recognizing that, is that these areas also provide critical climate change mitigation and adaptation benefits. So what I mean there is that they can help to sort of draw down atmospheric carbon dioxide, and at the same time provide adaptation benefits to the landscape. So it helps the landscape to adapt to, and the sort of communities that it support adapt to climate change. So things like helping to mitigate floods. It can help to provide thermal refugia for wildlife when we have extreme heat waves. It can help to temper the temperature of waters in the stream so that it is more habitable for organisms within. So many really important services that a healthy riparian area provides, or forest provides. And unfortunately, though, what we've seen is that over the past number of decades, that close to 100% of riparian habitat has either been lost or degraded in California. So a significant amount. And that what that degradation can be seen as, though, is sort of let me see, sorry, my slides are lagging. And I'm trying to go back right now. Okay, let's see. And so but what that offers is, that degradation is actually potential. And so riparian restoration can sort of bring back many of these services to the landscape, and realize that potential, that these areas can offer. And so this is just looking at sort of some of the communities that that riparian areas support. And so I really like this sort of habitat diagram on the right hand side, and all of the different bird species. This is for Central Coast habitat, or ecosystem, all of the bird species that have preferences for the different habitat types. So all the way from grassland down to the lowland areas and riparian areas. And you can see that the riparian areas actually support many species that depend on them. And so when you are able to restore them, it's in many cases, it's if you build it, they will come. And what we see here on the left hand side in this diagram is that is actually the case. So as you follow a riparian restoration over time, since it was restored, you see sort of linearly increase the number of bird species that are detected in that area. So that's, again, sort of really promising and showing that the importance of riparian restoration to bring back sort of the habitat, and then the critters that allow the critters who rely on that habitat to come back and thrive, hopefully. This is a busy slide, I apologize. Riparian restoration, it also can recapture carbon that has been lost from the land to the atmosphere. So if we just start on the left-hand side, this is a graph of the carbon dioxide levels in parts per million, and sort of years before today. So as you move left on it, you move farther away from today. And the current levels are, of course, here at that the highest point on this graph on the right-hand side. And a lot of that has come from burning of fossil fuels, but some of it's come from land use and land degradation, and from soils in particular. So lots from deforestation, and a lot from mismanagement of our agricultural lands. So, and that's sort of what this graph on the right-hand side is showing, is you have the land use under cropland and grazing land over time. And so the sort of the acreage that's under sort of cultivation or grazing. And then the yellow circles is the amount of soil carbon that's been lost concurrently. And so the amount of carbon that's been lost to the atmosphere from soils is as much as has been lost through global deforestation. So by basically removing a plant material globally, we've lost a similar amount, but from below ground. And together, that number is nothing to sneeze at. And again, though, what we can think about is sort of this degradation as being potential. And luckily, these systems, they cycle, right? Carbon cycles from different pools. It goes from the soils to the atmosphere to plants to soils. And so if we are able to restore these systems, allow the conditions for trees, for instance, to regrow along these riparian habitats, plant with plantings or not, that can start to draw down and capture, and if done at a scale, can potentially have significant effects towards statewide climate change mitigation goals, for instance, when combined with other land management activities. And so this is some work from one of my colleagues at Point Blue, Christy Diabala. She looked at a riparian area in the Central Valley, Cosumnes River, and she was looked at bird density and as well as carbon stocks, both above ground in trees and below ground in soils, and showed that with planted and naturally regenerated areas, you have an increase in bird density with that restoration, and you also have a significant amount of carbon gains above and below ground. So this is showing some data from California that indeed, it not only brings back sort of habitat for wildlife, but that it is building carbon at rates that are pretty high. So 3 to 4 megagrams of carbon per hectare is quite a bit. So again, potential there from that climate change mitigation potential, but then also when you, and we'll talk about this in a second, when you rebuild soil carbon, that actually has an adaptation component to it as well. So this is more work by Christy. She paired sort of her local analysis with another recent paper where she summarized literature from across the globe. And you can see the map on the left-hand side showing areas where that had studied biomass carbon accumulation in trees or in the soil with riparian restoration. And on the right-hand side is a graph showing stocks. So how much carbon is in plant biomass per area. So in this case, it's per hectare. In studies that she included in this review, this meta-analysis. So, and those are all in the dark gray. So these are riparian forests from across different sort of biomes or climate zones. And I think the important takeaway from this, from my perspective, is that riparian forests on an acreage basis harbor just as much carbon as other really critical sort of forest habitats we think about globally. So if you just look at those top two bars, you have riparian forest, with about 150 megagrams of carbon in above ground biomass per hectare, with tropical forests falling below that. So tropical forests, at about, I don't know if I eyeball it, 125 megagrams of carbon. So a considerable amount of carbon can be accrued on, again, this sort of area per area basis in these riparian forest habitats. And again, this is just from if you look at the graph on the bottom, it is just further demonstration that also translates to increases in below ground carbon in soils as well. So upwards of a 4% increase in soil carbon per year. So that's directly related to climate change mitigation, again, drawing down that carbon dioxide from the atmosphere. And if you scale it up with other management practices that do the same, the idea is that we can have an influence on climate, helping to slow climate change. And we know that land-based stewardship is a really critical piece of the picture, in addition to reducing and stopping emissions. And at the same time, it, especially when you're rebuilding soil carbon, it comes with so many other benefits. So soil carbon is like a central metric for soil health. It offers an incredible amount of services. So when we think about soil carbon makes up about half of soil organic matter. And so really soil carbon in this sense is an indication of how much organic matter you have in the soil. And that organic matter, what is the rest of it made up of? If 50% of it is carbon, well, some of it's made up of plant nutrients. So you have nitrogen, phosphorus, potassium, all of these other micronutrients too that plants need. And as that soil organic matter turns over, it supplies nutrients to plants. And so the more soil organic matter you have, the sort of slow release of nutrients over time. It also has, the way that I think about soil organic matter is that it has it can have these negative charges on it. And so, and the way I think about those negative charges is that they're like little arms that can hold on to nutrients. So it increases that, what we call cation exchange capacity. So it increases the ability of not only sort of supplying of nutrients over time, but sort of as nutrients are inputted, holding on to them through those extra arms. And same with water. That's why soil organic matter is also linked to increased water holding capacity. So as we think about adaptation to, let's say, drought, the more soil organic matter you have, the more water you could potentially be holding on that from what enters into your soil. And that's, of course, really important in those scenarios. So sort of when you think about all of these services, because there's more. You can have increased organic matter helping to reduce erosion. And so there's water quality as well implications. And so if you look on the right-hand side of this diagram, it's sort of showing you what are the outcomes of stewarding soil organic matter. And these include, sort of at the very end extreme, the no hunger, good health and well-being, life on land, climate action, clean water and sanitation, really big, important buckets that, of course, take a lot, are very multifaceted and nuanced, and soil organic matter, what this is trying to illustrate, does play a really central role. So if we can rebuild some of it through riparian restoration, that's a win-win in a lot of different ways, both for wildlife habitat and for soil health and climate change mitigation. Now, Point Blue has, many of you may be familiar with our Students and Teachers Restoring a Watershed program. I am not the person to tell the history of it, unfortunately, because it's a beautiful history. But I can say that the program has been around since the early 90s, and that we have focused on restoring watersheds, exactly what it's called, and including education and community engagement and outreach to revegetate, provide erosion control, invasive plant control, and project design and management for riparian restoration and restoration of other habitat types. And it's a really, really cool model. It's super fun. It's very impactful. And so our straw staff work through many partnerships with like local resource conservation districts and others, have had the opportunity to engage close to 50,000 students since their inception, working with almost as many volunteers, completing close to 800 restorations. And these numbers may be a little bit outdated, planting over 60,000 native plants and creating over 20 miles or 660 acres of restored habitat. So really recognizing early on the benefits of riparian habitats, the need to help restore them given their loss on the landscape, and taking action in what I think is a really meaningful way with partners to restore these areas in collaboration with students and teachers. So if folks aren't familiar with how this works, our straw staff coordinate with teachers to not only sort of do education in many cases prior to coming out and restoring sites, but then at the sites themselves are doing professional grade restoration projects with students I've been out there. I've had the pleasure of being out there a couple of times with, I think they were third graders. They were very young, all the way up through high school. So really cool opportunities, and have focused on the Bay Area. So we've had, a long history of sort of being in the business of conducting riparian restorations, given all of the benefits that it has. And more recently, led by some of the work that me and some other colleagues are working on, we have gotten in the business of quantifying carbon and soil health benefits of riparian restoration across California. So while we know that benefits are there and they're going to be there, and especially for riparian restoration, I think there's more certainty around that than for some other land management practices that are going on the ground for similar outcomes. That said, there's I think more we have to learn in terms of how much and how fast do we see some of these benefits, especially when we're talking about climate change mitigation, how much carbon is being built into the biomass, put into the soil, taken out of the atmosphere, that kind of thing. And then what does that mean for soil health? And so there's, I won't get too much into the weeds, but in terms of soil organic matter, remember I said soil carbon is about 50% of soil organic matter. Well, soil organic matter is not all made the same. And some soil organic matter has properties that allow it to sort of preferentially absorb onto soil particles and become what we call mineral associated. And that mineral associated organic matter is going to stay in the soil a whole lot longer, thus contributing to something like climate change mitigation than other kinds of organic matter that are in the soil. So these are called things like particulate organic matter. Those are going to turn over faster, but what's important about that is as it turns over, it releases those nutrients to plants. So both pools are important, but they serve different purposes. And it's important for us to understand how these practices are influencing those different components of soil organic matter. And so we are leading a big statewide study right now, taking advantage, working with partners to leverage the hard work that has already been done across many of California's riparian areas to plant native trees, restore these habitats, and see what the benefits have been. We're looking at areas that have been restored in the last 20 years. And so we're assessing in many cases projects that our straw team has put on the ground, but there are a fair number that were led by others as well. And so it's really neat to be able to go out there and try to contribute to the dialogue in that way. We also are, I'll just do a plug too, we just are sort of tying up as I speak, the what we're calling the Range C Monitoring Handbook. So this is essentially a set of protocols that dozens and dozens of practitioners and scientists have come together to create that are meant to be accessible, but also rigorous, and can help us help landowners, land stewards monitor carbon themselves with the restoration of riparian areas and other management practices as well on rangelands, and help to understand over time how these practices are influencing the landscape. So you can imagine that if one rancher uses these protocols and collects data, and another one does as well, you can, with appropriate eyes towards confidentiality and data sharing agreements and all of that stuff, if there's agreement to compile it at the network scale, and you can start to understand how these practices influence carbon in a way that you wouldn't otherwise be able to if it was just sort of a single organization going out there trying to have boots on the ground. It's more decentralized. Folks are contributing to that monitoring in kind of like a citizen science way, and helping to create a really large scale data set of practice impact for riparian restorations and soil health. So that's exciting to see sort of coming to fruition right now. There's a lot more to be done, but excited to see it where it's at. So, so those are sort of some of the benefits and how we're trying to help sort of get the practices on the ground and understand, quantify the benefits of them as they are. And given that riparian restoration does have so many benefits, a next question is, how can we make these restored riparian habitats resilient to climate change? So climate change is, of course, going to put stressors on the landscape. And when we're putting in lots of money into restoring these habitats, and there's so many benefits that are sort of at stake, what can we do to ensure that the hard work results in the outcomes that we want? So just as an example, in the San Francisco Bay Area, right, we're looking at more frequent and severe extreme events. So prolonged drought followed by really heavy rainfall. We're already seeing this, right? The wildfires, reduced surface water supply. There's going to be increased demand for groundwater. With warmer temperatures, you're going to have increased stream temperatures, and that's going to really, all of these are going to amplify the need for riparian restoration, again, to provide those refugia for wildlife, to move along corridors, to buffer temperature changes in the water, to help draw down carbon, hold water, mitigate floods. All of the needs for it are amplified, and at the very same time, those pressures are going to make it harder for the restoration to thrive, to be successful in the long term, because these are plant stressors. So what do we do? Well, one thing that we at Point Blue do is we take a climate smart restoration approach. And what we mean by that is that we're engaging in a process to enhance ecological conditions through our restoration in a way that makes them more resilient to the consequences of climate change, to those stressors. And we do that in by going through a number of sort of, or keeping an eye on a number of different climate smart restoration principles. And they're listed right here. So show your work. This is having to do with the fact that you're going to be looking at models in this process. You're going to be making decisions and sort of documenting what the decisions you are making based on the information at hand is going to be really important to know what in the future, if you need to go back and change your assumptions or your decisions based on as new information becomes available, for instance, or for others to be able to evaluate your work. And if they have to make sort of decisions based on your work in the future as new information becomes available, they're able to do that. Looking forward, but don't ignore the past. So the past can tell us a lot. What species have done well under different conditions? Are there particular populations within a species that have done really well? Who is tolerant of what, right? What species are tolerant of different conditions? We know that from the past, but then we need to look forward to see what the conditions at a given site are going to be in order for us to then be able to carefully select the species or planting palette that we want to introduce and think that will be successful. Keeping things local, generally local, but within that, looking at sort of local variability and genetic variability, both inter and interspecies variability, to create sort of a palette that will be as successful as it can be given future projected conditions at a site. Considering the broader context, so this is an example of this is thinking about where you restore. If these areas are really important riparian corridors, can you strategically create connectivity on a broader landscape scale by choosing particular areas to restore in relation to one another? So considering the broader context, building ecological insurance or functional redundancy is something I'll talk about just a little bit more in the next slide, and same with building evolutionary resilience, including the human community. I mean, education and buy-in and interaction from the community is critical to the continued adoption of climate smart practices. And so that's central, and the success of those practices. And so that's sits central here as well. And then research and monitoring. Of course, we always have more to learn. And so at Point Blue, again, we often take an implementation plus monitoring or science approach where they go hand in hand, so we can learn by doing. So the ecological, I'm just going to touch on these two a little bit more. The ecological insurance or redundancy is the idea that you would plant redundant species that have been collected from particular locations in the area or by species. And what you're doing there is similar to what an airplane would have, where you have maybe 4 engines. If one goes out, if one doesn't survive, you have others there to function similarly. So it's building in that redundancy that will help provide success in whatever ecological function you're trying to achieve, whether that's maybe food or habitat for pollinators at a given year, or a given time of year, sorry. So it could be any sort of function or service. If you build redundancy into that, then that will help to ensure that there will be winners, depending on wherever you land in that uncertain future that we have. Evolutionary resilience is, considering what your local site conditions are going to be. It may be wise to choose, whether it's acorns or nursery stock from locations that are currently experiencing different climates. And that may just be sort of microclimates within a watershed even. It doesn't have to, depending on sort of your comfort level with extending that boundary of where you're getting those stocks from. You can still build in, even if it's interspecies, intraspecies, so within a species, genetic variation. And so some individuals may have traits that allow them to withstand cooler, warmer, drier, hotter temperatures. And when you increase that diversity, again, what you're doing is you're sort of setting the stage so that there will be some that have the traits that allow them to withstand conditions in what is an uncertain future. We have predictions, but sometimes the predictions, vary widely. And so if you can build in that diversity, then you're going to ensure that the idea is there that you'll help to ensure that there's going to be some individuals who will be doing well in whatever the future looks like. So some key steps to doing climate smart restoration is, like many projects, starts with defining the objectives. What are you restoring for? Getting climate projections. So then using something like CalAdapt to identify what your climate is going to be like in the future, paying attention to things that might affect plants, like drought or extreme heat, climate water deficit, groundwater, that kind of thing. And then thinking about how the projected climate at that site will put your objectives at risk. So what aspects of your project will be put at risk by increased drought, for instance? Will that affect survival? And then you can go from there to say, okay, well, if there's going to be increased fire or increased extreme heat events, you can start thinking about what traits within your plant community do you want to try to ensure are there, and what tactics or principles can help to ensure and buffer sort of your project from those risks so that you ultimately have greater success. Point Blue does have a climate smart restoration toolkit. So there's a number of different tools within here. They're all listed right here that are meant to help folks choose, for instance, planting palettes, and have guides to designing some of the aspects of some of these projects. So I encourage you to check out those if you are interested. And just as an example, here's one. This is a climate smart riparian planting tool. And what it is it's a database of all the plants that might be considered for a planting project. And it has a list of their traits. So does it tolerate wet conditions? Does it tolerate dry conditions? Is it fire adapted? And then we also have lists of other traits as well. So again, like resources for wildlife. And you can start to, you can imagine how you'd be able to start to pick a planting palette where then you can build in redundancy, you can build in diversity, and you can start to address then, and you can do that with sort of an eye towards what your climate projections are at your site, and start to hopefully build a palette that will be as successful as possible. Okay, so I wanted to move on and talk about how we can harness the power of fungi to increase climate change resilience. So this is some recent work that I led in collaboration with some others at Point Blue. And the idea here is that can we build on our climate smart restoration principles and approach to consider the below ground community? And organisms below ground, fungi, bacteria, other organisms play a critical role in the functioning of healthy ecosystems. Many people have probably heard this statistic, but in a handful of soil, you have more organisms than there are people on the entire planet. And they are doing incredible work, much of which, we don't know specifically who is doing what, but some of them we do. And we generally know that they are very, very important. And so the sort of components or organisms within the soil microbial community that I want to highlight today are the mycorrhizal fungi. And so these, of course, are fungi that form important mutualisms with plants, where plants give those fungi carbon in exchange for nutrients and water. And so the fungi are able to they form associations with the roots of plants, and they can extend the rooting system effectively. They can get into spaces that plant roots might not be able to get. And so they can increase the amount of resources that the plant has access to considerably. And you can imagine under low nutrient conditions, or in the case of climate change, droughts, that increased access to water and nutrients so that they can photosynthesize efficiently is going to be really, really important. And there's a couple of different kinds. Well, there's a number of different kinds of mycorrhizal fungi, two commonly abundant ones. You have arbuscular mycorrhizal fungi and you have ectomycorrhizal fungi. There are others, but I'm just going to talk about those two today. And here in this picture on the left-hand side, it says endomycorrhizal. That's the same as arbuscular. So they penetrate plant root cells. They form associations with the majority of herbaceous plants. So your grasslands, lots of woody species as well too. Ectomycorrhizal fungi, they do not penetrate plant root cells. They go in between cells. And they tend to form associations with like a narrower selection of trees. So for California's rangelands, these are oaks, willows, alders, Doug firs, and importantly, when you're restoring a lot of these areas that have been degraded and devoid of trees for decades, those ectomycorrhizal species they can hang out in soil for a while, like as spores. They're able to, create some sort of dormancy, but they might be in very low numbers or absent at sites that have been degraded and absent of trees for a very, very long time. So when you're introducing trees then back to a site, if you are an oak, or if you are a willow, or if you are an alder or Doug fir, you may not have those mycorrhizal mutualists there in the way that you might want or would benefit from. And again, because these mutualists give them water and nutrients, if we want these restorations to thrive in increasing droughty conditions, for instance, one might expect that introducing the mycorrhizal fungi, the ectomycorrhizal fungi, would be beneficial and could be worthwhile. All sort of the trees and shrubby species that associate with arbuscular mycorrhizal fungi are, in my opinion, not a target for similar kinds of restoration. So what we'll call inoculation. Because they are there forming associations with all of the grasses. And so if you plant trees with that like to form associations with arbuscular mycorrhizal fungi, they're probably going to find a decent amount of fungi to form those associations with, since they're already there hanging out with the herbaceous plant community. Similar. Okay, so inoculation with microorganisms is not a novel idea. It's been done for decades. I think lots of people understand sort of the expected benefits and then also potential trade-offs of that. What is more I would say novel is this idea of harnessing and thinking about genotypes or genetic traits of the fungi that can confer additional stress tolerance to the plants. And so Another way to say that is these organisms, these fungi, just like plants, they can have climate envelopes. They can have tolerances or preferences for different conditions. So some fungi who have been hanging out in, a regionally hotter area through evolution will probably have traits that allow it to tolerate those warmer, drier conditions compared to its cousin over, in a wetter, drier a wetter, cooler area who's evolved for those conditions. They will probably have traits been selected for to have traits that help it thrive in those conditions. So if you, there's this sort of emerging interest in understanding if you can collect or condition soil communities to have stress tolerance for droughts or other stressors, and then they're forming associations with plants, do they confer that stress tolerance to the plants? So if those fungi are able, under future conditions, projected conditions at a site, at a restoration site, if they are have the traits that will allow them to do better under those, the assumption is, or the hypothesis would be, that then they can thrive and continue to give more water, more nutrients to the plant, as opposed to a fungal community or fungi that is not as tolerant or adapted. And so they might be struggling as well under those hot, dry conditions, and they might not be able to confer the benefits of that mutualism to the plant as much. So we were interested in understanding whether or not we actually saw some of those signals come out between sort of locally, like local fungi, mycorrhizal fungi, and they're still local, but locally, but hotter and drier fungi, and whether or not that could help increase near-term success of some of these straw restoration projects. So we generated an experiment, a field experiment, to test this out. And our two questions were whether or not whole soil inoculum could increase plant success and vigor in the near term. Again, just looking at these ectomycorrhizal host species. And then, does inoculum from warmer, drier climates in the region do better? Does it increase that plant survival and vigor compared to the local inoculum or no control at all? So we had 3 riparian restoration sites and 4 plant species. So we had we had coast live oak, valley oak, and then we had Doug fir, and we had a royal willow. And we had 3 inoculum treatments. We did local, which was that like sort of regular climate, so it had a climate similar to the restoration site and projected. And then we had a climate adapted, where we collected inoculum from areas that currently experience the projected climate from the restoration site, and then a control or no inoculum. And I hopefully, I think we're doing okay on time. I was going to play a video, and then this is going to take us close to the end. It's a just over 5 minutes, I believe, and it's a video we put together sort of highlighting the process of this inoculant-supported restoration and talking through the results. And I thought it might be nice to show it here. So I'm going to go ahead and play it, and everybody enjoy, and then we'll talk more when it's done.
Hi, I'm Chelsea Carey. At Point Blue Conservation Science, I serve as the Working Lands Research Director and Principal Soil Ecologist. In this video, I'm going to talk about an inoculant-supported restoration project that Point Blue recently undertook and provide guidance on how to conduct this kind of restoration yourself, should you be interested. Healthy riparian forests provide critical services to humans and wildlife. These include clean water, protection from flooding, wildlife habitat and refugia, and climate change mitigation. Unfortunately, the majority of riparian forest habitats across California have been lost or degraded. At Point Blue, we have a long history of restoring riparian forests throughout the Bay Area as part of our Students and Teachers Restoring a Watershed program. In this program, we work with kids of all ages to conduct professional restoration grade projects that include planting native trees and shrubs to rebuild the health of these important areas. We take a climate smart approach to our restoration projects, which means that we make sure we plant a diverse array of local species and that we include ones that have ecological traits that help them to withstand projected conditions at a site. This might include hotter and drier conditions or more frequent fires. Inoculant-supported restoration builds off our climate smart approach by also considering the life below ground. In a handful of soil, there are more organisms than there are people on the planet. And some of these organisms form really important partnerships with plants. Mycorrhizal fungi in particular form associations with plant roots and help the plants get the water and nutrients that they need to grow. For some of the plants that are common to restorations, like oaks and willows, the mycorrhizal fungi that these plants form associations with, which are called ectomycorrhizal fungi, they may be absent or at low densities at those sites. This is especially true for sites that have been devoid of ectomycorrhizal host trees for a long time. We found that introducing whole soil inoculum that includes mycorrhizal fungi during the restoration process helped to increase germination and early survival of both coast live oak and valley oak trees. And in fact, introducing inoculum that was collected from local areas that are warmer and drier than the restoration site itself, that actually amplified the benefits seen for coast live oak and arroyo willows. Specifically, the odds of survival with heat-adapted inoculum were 4 times higher than controls for willows and 3 times higher for coast live oak. One possible reason for this is that the fungi from warmer and drier regions have traits that allow them to withstand drought or higher temperatures in the areas that they were introduced to, thus conferring greater benefits to the plant. The process of conducting inoculant-supported restoration, the way that we've done it and recommend here in this video, it includes 4 steps. In this video, I'm going to walk us generally through these 4 steps, and I encourage you to check out the companion technical report guide for more details. The first step is to identify sites from which you'd like to collect inoculum. Using local knowledge or GIS and publicly available data sets on climate and soil type, find a site that fits your criteria. In our project, we were looking for collection sites that had the target tree species of interest, had similar soil, and either similar climate or warmer and drier conditions compared to the site that was being restored. Once you've identified a location from which to collect inoculum, the next step is to make a site visit. You'll want to find 3 to 5 individual trees per host species. So in this case, we sought out coast live oak, valley oak, arroyo willow, and Doug fir. Focus in on those individuals that don't show any obvious signs of disease and which otherwise appear to be doing all right. At each tree, you'll collect soil twice, once upslope and once downslope. You'll go ahead and take 1 pace away from the tree's trunk and use a pre-sterilized hand trowel or soil knife to collect soil from the surface down to 15 centimeters depth. You'll do this excavating a hole that is about 10 by 10 centimeters in area. Go ahead and combine the samples in a gallon-sized Ziploc bag, keeping samples from each tree separate. You can transport those back to your facilities for storage and Phytophthora testing. Now that you've collected your soil samples, you're ready to do Phytophthora testing. Phytophthora are pathogens that are responsible for killing plants throughout California. Perhaps most conspicuously, they're the culprits behind sudden oak death. Phytophthora species can spread to new sites during restoration. At Point Blue, we take every precaution to minimize the possibility of that happening, and we encourage you to do the same. A relatively simple yet effective way to test for the presence of these pathogens in plant and soil samples is to conduct a method we call pair baiting. We've adapted the testing protocol we use from an online source called Phytosphere. The basic approach here is to use green pears, Danjou pears preferably, to bait any Phytophthora that may be present in your sample and to watch for signs of disease. This entails first reviving dormant Phytophthora if necessary by moistening soil with water for a few days. Then baiting the pear by incubating it in your soil sample that has been saturated with water to the point of flooding. Be sure to mark any existing blemishes on the pear prior to addition to the soil. After about an hour, drain the leachate from the soil and continue incubating the pear in the leachate for 3 days. At the end of the 3rd day, remove the pear from the leachate and let sit for 5 more days. All the while, keep an eye out for signs of Phytophthora. You'll want to report and discard any samples that show symptoms and save the soil and leachate from those samples that were clear. These can be stored at room temperature and used as inoculant during the restoration process. In this portion of the video, I'll be joined by Jesse Ditmore, a straw project manager with Point Blue. How you install your plant is going to depend on what kind it is. For instance, planting willows typically occurs by inserting cut sprigs, whereas planting oaks is often done by burying acorns. Regardless of the plant species, the method to add whole soil inoculum, it's going to be similar. And this includes adding soil and leachate from pair baiting to the bottom of the planting hole. Because you're simply trying to introduce enough ectomycorrhizal fungi to colonize the plant roots, we found you don't have to introduce much inoculant. In our study, we added 2 tablespoons of soil and 1 tablespoon of leachate with each plant. You can complete your plant installations as you normally would, but now they have their added microbial partners to help them survive better in the near term and hopefully thrive in the long term. If you'd like to know more about how to conduct inoculant-supported restoration, again, I encourage you to check out our technical brief. I wish you all the best as you endeavor to perform inoculant-supported restoration as part of your next restoration project.
Awesome. Okay, so hopefully everybody's still with me. That video is, let's see, how do I get off this page now? Okay, that video and the technical report and another sort of like a handout, a 2-pager, can be found at Point Blue's website at that link right there. And I won't spend too much more time talking through the results since we did mention it since I mentioned it in the video. But this is that graph, and and I just want to highlight that we did find strong evidence that drought tolerant inoculum individuals that were planted with drought tolerant inoculum more than tripled the odds of survival in arroyo, willow, and coast live oak. It also the odds of acorn germination doubled for both oaks. The effects of treatment with local soil inoculum were less statistically significant in many cases, but it did improve the germination rates of valley oak. And so we thought that these results are interesting. They are compelling. It's something that we're going to continue exploring in-house at Point Blue, and have created the resources for others to explore if they would like. And I'll just end there by saying, I think my 3 take-homes from this talk are that riparian restoration, it's important. It's important for many things, including wildlife, providing habitat for wildlife, mitigating climate change, and providing climate change adaptation, building that into the landscape as well. And that we can build resilience into riparian areas by following climate smart restoration principles and designs, and that Point Blue has many resources on our website to help individuals do just that. And then explicit consideration of the below ground community could help to improve the success of restoration projects in the face of climate change. And so I'll end there. My email is right there. Anybody should feel free to follow up with me should they want. I'm also on Twitter, and I'm going to stop sharing my screen, and I think then we can open it up. I don't know how much time we have to Q&A.
Thank you so much,
Yeah.
Jesse. That was fun. Really interesting. And
Thanks.
there are a few questions, of course.
Awesome. Okay.
But so if you've got some time, I mean, there's a couple questions. There's not like a ton. That was a lot of information. So
yeah. Okay. Yeah. So should I just kind of look through do you have them?
Yeah, we're happy to do that for you if you like.
Okay, sure.
I think the first question was from Carey. She asked if regarding inoculating restoration sites with more vigorous, resilient MRF, is there any concern about introducing an organism not native to the area being restored? I mean, other than the inoculant.
Yeah. I mean, certainly, I think, so we definitely don't want to introduce pathogens, and at Point Blue, we take that very seriously. So that's why we do the Phytophthora testing. The Phytophthora testing also does show that pears are sensitive to Pythium, which is another pathogen. And so we will tend to disregard samples that have a lot of Pythium in them too. And so definitely trying to, dot our I's and cross our T's and encourage others to do the same for those really nasty pathogens. In terms of other organisms, yeah, it's interesting. It's hard to know. So we do stay regional. We're not trying to go very, very far. Microbes do disperse. So I think a lot of that might be surprising to folks, but microorganisms disperse globally. They are dispersal limited. So there is patterns like across space and time. Not everything is everywhere. So certainly you could be introducing organisms that are like non-native to a site, but it's really hard to know what's native and non-native for microorganisms. And so I think I'm personally comfortable with sticking regionally and moving organisms. I mean, many people go as far as, doing commercial inoculum, which is definitely going to be even more non-native. And so I think it is a little bit of sort of a comfort level and preference almost. And I don't know if there's a right answer. It's definitely something to keep an eye towards and to study, for us to be able to study more, which we can now that we have really cool molecular techniques that we can look at the community in new ways. And certainly don't want to be moving soil around all willy-nilly because we know microbes can be invasive. I mean, that's what sickness is, right? It's a microbe invading your body. And so, yeah, but I would say if you stick regionally and if you take the proper precautions to make sure you're not moving around pathogens, those are really good first steps.
Yeah. So Ellie asked also, is going to Southern California too far?
I don't know. I mean, there's no right answer. I would probably, if it's me, I would try to stay like more regionally, like locally. So within, the county, for instance, or within a couple of counties. So, within the greater Bay Area, that kind of thing.
Yeah, I mean, there's probably quite a few people in this group that are kind of like us. We're a little hypersensitive to introductions.
Totally, yeah.
I mean, San Francisco is like the introduction capital of the country, probably, and so many things will grow here. So it's kind of like, okay, there's places for plants from all over the world. And so there's been in the horticulture industry, there's been a push to say, well, with climate change, we need to bring plants from Southern California up here. And we're like, whoa, wait a minute. That's where, I don't think there's much evidence that we're seeing, we're not seeing our native areas here locally along the coast anyway, suffering any more than any other, groups from the dry climate. And which brings me to a question of, during this experiment time that you tested out those inoculants, was that all during the drought period we're in now, or?
It was 2018, is that right? 2018, 19, or 2019 and 20. And it was right at the start of the pandemic, actually. So when was that?
Okay.
And that is important because what that meant for our restoration projects is that they didn't get as much maintenance. And so I believe that they probably were a little bit more stressed than normal because they didn't have the normal watering and maintenance and as they would. And, I think, like the silver lining of that is that it is sort of initially stressed and was able to, for us to really see what the inoculants do under those immediate sort of, more stressful conditions.
Right. We're of course all hoping that we may not be in a drought, forever.
Yeah, exactly. That is definitely,
it seems to be a trend, but we've also seen droughts go away here as well.
Mm-hmm.
Yeah. So Jean asked about a little more specific about the pair testing. I think, that was a really fast, of course, we are recording this, Jean, so you can go back and watch it in slow-mo. But anyway, is there some place where we can get information on that, where Gene can go and get information on that technique?
Yeah, so the video itself is and then it also has a technical report which walks through all those steps, is available on Point Blue's website. So I think it's pointblue.org/ISR. The pair baiting method that we use comes from, I think the website is Phytosphere. And so you could go to Phytosphere for more, like a lot of detailed information on pair baiting and Phytophthora testing.
Oh, cool. Carey Olson just put that very thing in the chat there.
Awesome. Yeah.
Yeah. So, well, hey, we're gonna let you go. We know you've got a 3-year-old dozing.
Hopefully dozing.
Hopefully
Yeah.
dozing. Listening in, learning a lot.
Yeah.
Thank you so much. We really appreciate you sharing your research and time with us tonight.
Yeah, thank you so much. Thanks everyone for joining and for spending the evening with me. It was, yeah, it's fun to talk about this all with you, and follow up if there's additional questions or thoughts. Yeah, you have my email.
Will do.
We'll see you up there at Point Blue.
That's right. See
All
y'all.
right.
Thank you.
Bye, everybody. Thanks for being here. Good night.
Bye.
Good night.